high-throughput sequencing. So please, Amy, go ahead.
Thanks so much, Alex. And before I jump in, I just want to give two thank yous. The first one to all of the organizers. I think it just takes so much work to put on a conference like this, especially in this online format. So I really appreciate all of the work that they've all put in to make this possible. And I also really want to thank all of the speakers and panelists from this morning. There were some incredible talks that were really interesting, really clear, really polished, really professional. And so it's really a pleasure to be a part of this terrific session. So thank you for having me. As Alex mentioned, I'm Amy Willis. I'm an assistant professor at the University of Washington. And I'll just jump straight into this presentation. I won't be monitoring the chat while I talk, but at the end of when I finish presenting, I'll come back and I would love to take some questions. So we've heard a lot this morning about high-throughput sequencing and how high-throughput sequencing allows larger scale data generation in a cheaper and more efficient manner than ever. And it's totally revolutionized the way that we study biology and medicine and microbiology. It's totally changed the way that we perceive and understand microbial ecosystems and microbial interactions. So one thing that a lot of folks spoke about this morning was using long read and perhaps or short read or both technologies to figure out who's in the community in terms of maybe metagenome assembled genomes and figuring out sort of what's present. So this talk is going to be focused on analyzing the abundance of microbial strains or taxa in a community. And I'm going to be focusing on essentially sort of the step that comes after we've figured out who is there and we start to sort of quantify their abundance through all of the rich information that high-throughput sequencing data provides us with. So as many of you know, the process of generating sort of downstream microbiome abundance tables requires a lot of steps, and some of those steps are performed in the wet lab and some of those happen sort of in the dry lab. So steps that are required in order to generate these relative abundance these abundance profiles include sample collection, sample handling, sample storage. We have to be getting at the DNA that's in these bacterial cells. We'll do that by lysis or DNA extraction. In some cases, we might be targeting a specific section of the genome, maybe the 16S gene. In other cases, we might be doing whole genome or shotgun sequencing. Depending on the biomass setting we're in, there might be a DNA amplification step. Then we take our product, a DNA product to our sequencer, maybe that's nanopore, PacBio, Illumina sequencing, and then sort of fits in the step that we had a lot about this morning, this sort of grouping of sequences into units, maybe strains or genomes. And often what comes next is some form of counting the abundance of those sequences. So maybe that's mapping reads to our assembled genomes, maybe that's counting amplicon sequence variants for in marker gene setting. Maybe there'll be some sort of read recruitment step. But a really important question that I think has been underexplored by the microbiome literature to date is, what is the impact of all of these upstream steps on those resulting count profiles or relative abundance profiles that we get at the end of all of these choices that we have to make? And so a look at the literature really quickly reveals that all of these steps have an impact. And so sample storage and sample handling have an impact on resultant microbiome profiles. We have that DNA extraction, the way that we lyse the cells, can have an enormous impact on the profiles that we get. Whether or not we choose to do targeted marker gene sequencing or we choose to do whole genome sequencing impacts the profiles that we see, as well as amplification, whether or not we choose to do DNA amplification through some PCR process, and also the chemistry of the sequencing platform that we end up using. And so if all of these steps have an impact, what is the ultimately what can we do with the data that they generate? And so all of this work, of which, and there are hundreds more papers, this is not an exhaustive list, hundreds more papers on the subject of what is the impact of upstream lab choices on resultant microbiome profiles, as well as, of course, bioinformatics choices, such as were discussed this morning. What is the impact on the resultant profiles? And that literature has primarily focused on variation between different handling protocols or extraction protocols, amplification protocols. but there hasn't, in my feeling, there hasn't been a big emphasis on the accuracy. And so what we have ended up with as the result of this literature is this current paradigm where it's widely believed that we can compare quantitative data such as relative abundances or counts of sequences from either shotgun or 16S sequencing across samples within the context of a single study for a set of samples that have all been sequenced together and sample prepped together, but we can't compare that quantitative data across studies because we know that all of these choices upstream have an impact on the downstream profiles that we're going to be analyzing. And so a question that's left open in my mind, and the one that I want to talk about today, is kind of an obvious one, and it's how accurate are those quantitative microbiome data profiles that are the result of all of these different steps. And so not so much in the sense of how precise are they, how much do they vary between all these different choices, but what are they actually telling us about the microbial communities that data was generated from? What are they telling us about the biology, or to what extent are they just telling us about all of these lab choices that were made that are sort of artifactual as a result of the data measurement process? So everything that I'm going to talk about today applies to both marker gene sequencing and also shotgun sequencing. Specifically, I'm going to be talking about taxonomic profiling, and so abundances of genomes or amplicon sequence variants, and all of that data can be expressed. All of that data can be expressed in counts. It can be expressed as relative abundance. It can also be expressed as something like coverage, and I don't have an example of what that data structure might look like here. So this work has been published. I'm lucky to work with some terrific collaborators. So I just want to thank Michael McLaren and Ben Callahan, who are both at NC State and who are co-authors of this paper, which was published last year in eLife. So it's consistent and correctable bias in metagenomic sequencing experiments. So feel free to take a look. And I'm also happy to answer questions. Okay, so how would we go about evaluating the accuracy of relative abundance data or microbial abundance data? And so a natural tool that we have at our disposal is looking at mock communities. So many folks have talked about analysis of mock community data earlier today, but for those who aren't familiar with them, mock communities are sort of artificially constructed microbial communities. We often build these to benchmark sequencing protocols as well as bioinformatics pipelines. And the reason we do that is because the composition of the community is known. So we know who's in there, and we can also construct them in such a way that we know the abundances or the ratios of taxa to each other in these artificially constructed communities. And so we're going to look at some data from Brooks and co-authors that was published in 2015 in BMC Microbiology. And so the Brooks data is very interesting because they constructed about 82 different mixtures of 7 bacterial taxa, which are given in the top right here, in equal abundances. So all taxa are equally present in all samples, but there may be only a mixture of 2 or 3 or 4 or 7 taxa per community. And so what I've shown here are bar plots from what we know is present in 8 of the samples out of the total of 82. And so if we had technology that allowed us to perfectly accurately see the relative or assess the relative abundances of taxa in the communities, our observed relative abundance profiles would look exactly like these true relative abundance profiles. But what we end up seeing in terms of the relative abundance profiles at the end of sequencing looks like this. So we can very quickly see that something's going on in our observed data that makes it not representative of what taxa are or the abundances of the taxa that are present in the community. So we tend to be able to see who is there, but we are way off in terms of determining their relative abundances. So this is an accuracy issue of sample relative abundance profiles for true relative abundance. So this particular data that I'm showing is 16S sequencing. And so a really natural question that a lot of folks ask is, well, do all of these taxa have the same copy number? And could copy number variation be accounting for the distortion between true and observed relative abundance profiles? So here's the true abundances of the taxa that a way that's been adjusted for variable copy number across these organisms. And we see that maybe it fixes, maybe it sends us further in the direction of the truth, but it's certainly not accounting for all of the deviations between observed and actual relative abundances, even copy number adjusted in our community. Okay, so just to really hammer that point home that observed relative abundances are not good estimates of true relative abundances, even for simple artificially constructed microbial communities, we can see that we have almost no correlation between true and observed relative abundances. But if we start to look a little more closely, we see maybe some more consistent patterns. So we tend to see that G. vaginalis and S. agalactiae tend to be under-observed relative to their true relative abundance, and maybe some of the other taxa tend to be over-observed relative to their true relative abundances. So what we're going to do, what we did is we took a bit of a deeper dive and said, well, obviously, in general, we have little correlation between true and observed relative abundances, but is there something that is consistent sample to sample that we could maybe use as a target for analysis? And this is going to have impacts on how we should be analyzing microbial communities, even not mock communities. Okay, so I've pulled out 5 samples, some of which were shown on the previous slide, and I've shown the true relative abundance, not copy number adjusted, and the observed relative abundance profiles that we have. And what we see, if we look closely, is that we tend to see that this green taxa, which I believe is Lactobacillus iners, tends to be over-observed compared to this blue taxon. You'll notice that all the green and blue taxa are present in every single one of these samples. And so what if we have a look at the ratio of the green taxa to the blue taxa across all of our samples? And so what we see when we do that is that Lactobacillus iners tends to be observed about 20 times or 18 times more often than S. agalactiae, even when they're present in the same abundances. And so there's some variation about this sort of 18-fold over-observance of Lactobacillus iners compared to S. agalactiae, but it's actually pretty small, a standard deviation of about 1.5 compared to an effect size, mean of about 18.5. So that's encouraging and suggests that there's maybe something that is consistent about the relative abundance profiles, even when the remainder of the community differs across these samples. So a natural question to ask then is, well, is this a specific feature of only these 2 taxa, or is this a common feature across all pairs of taxa? And the answer is it tends to be, in this data at least, a very consistent feature across all pairs of taxa. So here's about 10 more different pairs of taxa, and we can see that the ratio of the one taxon to another, keeping in mind that their actual ratio of each other is 1:1, tends to be very consistent regardless of the other taxa that are in the mixture, and also the number of other taxa that are present in the mixture. So this figure is taken directly from McLaren and co-authors, the eLife paper that I mentioned earlier. Okay, so the next thing that we want to think about is, what does that suggest about the data generation process? What can these figures tell us about the connection between observed relative abundances and true relative abundance in such a way that may be able to help us understand what is consistent sample to sample in such a way as to make use of microbial relative abundance, microbial abundance data. Okay, so this data I think provides really good evidence for the following model, which is that observed relative abundances are biased estimates of true relative abundances. And the reason why they're biased is because every taxon has some specific efficiency factor that determines whether or not it's over-observed or under-observed relative to the other taxa in that mixture. And so if you want to lean into this notation, we have that the expected value of our counts, our WIJs, so that's our sample relative abundances, is equal to the true relative abundance of a taxon multiplied by its efficiency divided by essentially the normalization of the relative abundances of the other taxa in the community times their efficiencies. So we have a sample-specific denominator here and a taxon-specific efficiency factor here in the numerator, as well as what we actually care about, the underlying biology of the true relative abundances, Pij. Okay, so I want to talk a little about the implications of this model, because I don't want it to be dismissed as just some statistician's concern about models and model misspecification. The impact of these taxon-specific efficiencies are actually quite far-reaching and can lead us to make conclusions in the absolute wrong direction than we should be making from relative abundance data. Okay, so let's take a look at a specific example. So here I've shown 2 microbial communities observed without any noise. We've got a treatment sample and a control sample. Maybe we're looking at 2 different disease states. And what we see based on these observed relative abundance profiles is that the relative abundance of this orange taxon, arbitrary taxon, is lower in the treatment sample compared to the control sample. So what true data could have generated this observed profile? So here's the actual relative abundances that could have generated those observed relative abundances. So what we see is that in the actual underlying biology, what we really want to be measuring is that the orange taxon was actually in lower relative abundance than its relative abundance in the treatment sample. But in our observed data, the effect was actually the other way around. And so what's happening is that we have a complete flip in what we should be determining, what we'd like to be determining, compared to what we actually see from our relative abundance data. So how could this have happened? Here's the efficiency vector that I used to generate this data. So we see that, so I have that efficiencies are all calibrated relative to this navy blue taxon here. We have that the orange taxon is observed with 6 times greater efficiency than the blue taxon, and the green taxon is observed with 18 times greater efficiency compared to the blue taxon. And so if we use that model that I was describing earlier, where we have the product of the actual relative abundances and these taxon-specific efficiencies, and then this sample-specific normalization factor, we end up with this observed taxa on the right, and you're welcome to check my math if you'd like. So how do we conceptualize this enormous difference between the actual relative abundance profiles and the relative abundance profiles that we observe? So what's going on here is that the green taxon is a high efficiency taxon. It's much really over-detected essentially with it, compared to its actual abundance in the community, because it's got a much greater efficiency than the other taxa. And so the abundance of the green taxon increased between the from the control to the treatment group. And so in some sense, the green taxa sort of ate up more of the pie of what was available to be observed. Similarly, this blue taxon, which was in high abundance in the control sample, has very low efficiency. And so it sort of made space for that orange taxon to increase in abundance in the observed control data. And so a really important factor here is that the really important point that I want to make here is that the relative abundance of that orange taxon doesn't only depend on the actual relative abundance of the other taxa. It depends, and it doesn't only affect on the efficiencies of the taxa in the community. It depends on the abundance of the other taxa as well. So it doesn't just matter who's in the community, but also what is their actual abundance, which is also an unknown parameter in these models. And so I think a big takeaway from this message is that we cannot accurately estimate changes in relative abundances or in proportions based on data generated from this model, which we have good support for based on this Brooks data and also another data source that is described in the paper regarding shotgun data. I'll come to that in a second. So in contrast, if we can't estimate relative abundances in a reliable way, is there some other feature of these microbial communities that we can? And so what I want everyone to take a look at here is every sample's ratio of the orange taxon to the dark blue taxon, and then the ratio of that ratio in the treatment compared to the control sample. And we see that the actual ratio of ratios is 3.7, which is perfectly preserved in the observed data. Again, this is a noise-free setting. And so what this is telling us is that even if we can't estimate relative abundances or changes in relative abundances based on data generated from this model, we can accurately estimate changes in the community through ratios rather than through relative abundance data. So that, I think, provides some more concrete guidance about what we can do to move forward in the absence of knowing these efficiencies, which in general we don't. Okay, so all of this work and some other work supporting this, this model is discussed and is available in this paper published in eLife. So essentially, we validated this model whereby each taxon has some efficiency factor or some bias factor that confounds the estimation of relative abundance. So while we would like to be observing some unbiased estimate of the actual relative abundance of taxa in the community, we're observing some convolution of something we do care about, the underlying biology, and also some aspect of our measurement process, the sequencing steps that go in upstream. And so I won't talk about it today, but in that paper we validated this model both on 16S data and on shotgun metagenomic data. So please don't let me leave you with the impression that shotgun data is free of these biases. It turns out that DNA extraction or cell lysis plays a massive role in determining these efficiencies, which makes sense because the efficiency with which you can get DNA out of a cell is going to impact how much DNA you see from that cell. So take a look at the paper for some information about the shotgun analysis that we did. So another tool that's out there for estimating these efficiency factors, if that's what you care about, is to perform calibration with respect to some sort of gold standard tool. And so here the gold standard, even though we saw this morning, there's definitely some technical variation in this data as well, is taxon-specific qPCR data. And so essentially what we see is that the relative abundances based on 16S data look very different to relative abundances that we find based on higher quality qPCR data, which are subject to different biases. But what we can do is calibrate the 16S data to our taxon-specific qPCR data using a model similar to the one that I discussed earlier. So I think of this as a way of using cheap and high throughput data like 16S that has problems with the efficiencies with respect to which taxa are detected with this more expensive, more labor intensive, low throughput, but higher quality technology of taxon-specific qPCR primers. So if you're working in a, especially a clinical context, or your collaborators have clinical, care a lot about, have developed qPCR primers for a specific taxa, this method described in this paper that's available as a preprint may help you model or may help you predict the actual abundances of taxa in your community, even the ones for which you don't have qPCR primers. So one thing I just want to flag for this community, I'm trying to get this message out there, is that a lot of folks look at relative abundance data obtained from shotgun or 16S data and they say, oh, we saw 24% of reads mapped to Firmicutes genomes, and so therefore the relative abundance of Firmicutes genomes in our environment is 24%. So I really want to move away from that language. We do not, our relative abundance data is not representative of the communities that they're taken from. Another idea that's been floating around a lot is that we can take total 16S qPCR concentration, bacterial concentration estimates, and multiply them by broad range 16S relative abundances to estimate taxon-specific concentrations. That model totally falls apart in the presence of efficiencies, which can vary significantly across taxonomic and phylogenetic breadths. So I really would love folks to move away from that paradigm as well. Okay, so I'll wrap up with some implications to try to make time for a lot of questions. So I think the big implication is that relative abundance data can be misleading. And so a lot of folks think of shotgun or 16S data as compositional or relative abundance data, but in fact it's not. It's some convolution of actual relative abundance and artifacts as a result of the sequencing process, including DNA extraction, amplification, and sequencing. Preliminary results suggest that ratio-based analyses may be more reliable because we can reliably estimate ratios of taxa and ratios of taxa across different experimental treatments, perhaps with some model. And so there's ongoing methods work in this area to try to provide tools for the community for accurately determining changes in microbial communities as a result that can function under these varying efficiency models. One thing that I can direct you to the paper to take a look at is that efficiencies can vary widely across taxa, including taxa that are within the same genus, so different species within the same genus. And so as soon as you start aggregating relative abundances or aggregating counts up phylogenetic trees or up taxonomic trees, we start to estimate something that's even less like the actual relative abundance of taxa. And I would say one final thing, and it pains me, folks who've seen me talk before in the previous last few years will notice that it probably pains me a lot to say this, but quantitative diversity indices like Shannon diversity and Simpson diversity and Unifrac, which all take sample relative abundances as input, are not estimating something that is a component of the actual underlying community. They're estimating some confluence, again, of biology and sequencing. So I'm moving away from quantitative diversity indices because they're not estimating anything associated with it, or they're not estimating something associated with the community free from bias associated with sequencing. So I'm working hard on this ongoing work. We're looking for robust statistical methods to determine changes in microbial communities. We're looking at using this model to help you choose between different options that you have in the wet lab. So rather than aiming for a protocol that's unbiased, which we think is impractical and infeasible and is what's less biased is going to differ between different sample types, and there's never going to be a universal protocol. So how do you choose between them, particularly in conjunction with some methods analysis plan that you have? Similarly, how do we use positive and negative controls data to estimate these efficiencies? And the ultimate long-term goal is to be able to do calibration of biased 16S sequencing data to get unbiased relative abundance estimates from this data, and also to allow us to do meta-analysis, so comparing data across from multiple different studies, which would allow us to really do validation and, I would say, science. So so what I talked about today for folks who took a nap, I know they've got folks from many different time zones. I talked about a mathematical description of the way that relative abundance data becomes biased for, relative abundance data is biased for true abundances. So that applies to both 16S and shotgun data, whether or not you're looking at sequence variants or strains, whether you're looking at coverage to determine abundance or counts or relative abundance. This model applies to all of those equally. This model has quite serious implications for how we should be analyzing microbiome data. In particular, looking at proportions can lead us to conclusions that are not only wrong, but also wrong in the wrong direction. So we can look at I went through an example earlier where we saw that the relative abundance in the actual underlying communities decreased, but in the sample data, it increased as a result of these efficiency confounding features. And I'll say that some analysis methods are more robust to bias than others. For example, ratio-based analyses might be more robust than looking at relative abundance or proportion-based analyses. I work with some terrific people on this work. Michael McLaren and Ben Callahan at NC State are a real pleasure to work with, and both are brilliant. David Clausen at UW is doing pushing a lot of the methods work forward. So he's going to be named to watch, I think, in the next few years. And Jim Hughes and Brian Williamson, both at UW, and Brian Williamson's now at Fred Hutch, have also been fantastic collaborators for integrating 16S and qPCR data. I didn't have anything more to add, but I am really glad to be here, and I would be very happy to take questions, and thank you.
Yeah, great. Thank you very much, Amy.
My pleasure.
And there are already a couple of questions. Can you see the Q&A?
I am just closing my screen so that
Okay.
I might be able to. Okay. All right. So I'll just start from the top. Does that sound good?
Yes, sounds good.
Yeah. Okay. Had an anonymous question For most metagenomic analysis, there's approximately 50% unidentified microbes. If we do not know about the composition, how can we efficiently calculate the ratio? So that's a terrific question, and one that we receive a lot. I would say that the wrong or a much harder way to go about using these results is to say we need to try to estimate these efficiencies of all of these taxa. I think that's not going to be scalable. We have enormous microbial diversity. We've seen that very similar phylogenetic taxa that are very similar phylogenetically can have enormous changes, order of magnitude differences in their efficiency. It's just not going to be scalable to be quantifying or estimating the efficiencies of every taxa under every experimental protocol. And then you think about that DNA kit, extraction kit goes out of stock. How do you adapt? It changes entirely. So I would say that my personal view on where we should be going with this is not trying to estimate these ratios. Unidentified microbes, unculturable microbes, changes in the sequencing process, all are going to make this just unscalable. And so rather than calculating these ratios, I think what we should be looking towards are analyses that are more robust to the presence of these efficiencies, rather than actually trying to estimate the efficiencies themselves. I might jump into the next question, and happy to follow up with anyone who has further follow up on that. How robust are taxon-specific efficiencies across different experimental setups and bioinformatics pipelines? Are different pipelines expected to provide similar estimates in terms of efficiencies or abundance ratios? That's another great question. So one thing that we looked at in the paper is essentially at what steps of the experimental pipeline are these biases introduced? And so the Brooks data is very unique in the sense that they looked not only at live cell mixtures, but also at mixtures of extraction product and also PCR product. So you can see sort of what aspects of what elements of the protocol introduce greater and smaller biases of components of these efficiencies, because they multiply across these steps. Can folks still see my screen?
Yes, it's
yes,
a little
okay.
bit small, but yeah,
okay. I'll try to make that a little larger. And so this is a figure from the paper where we looked at, where essentially what we found is that the extraction step, the DNA extraction step, has the largest effect of the workflow across efficiencies. So maybe to go back to the original question, I would say that smaller deviations, for example, bioinformatics, maybe post-PCR, so yeah, the effect of sequencing chemistry, as well as, I guess this was maybe all Illumina. Different components are going to affect the resultant efficiencies differently. Preliminary results seems to be that extraction efficiency is one of the largest components. So I would say take especially a lot of care in interpreting data obtained from different DNA extraction protocols. I'm happy to follow up on that as well. Third question, how do I address the problem of taxa with zero read counts when calculating these ratios? Do I recommend zero imputation? Yeah, good question. And so I think I'll sort of answer this with, similarly to my response from the first question, I don't think that estimating these efficiencies is scalable. I think that looking at coming up with analyses that are more robust to this type of perturbation, systematic perturbation in our data, is going to be a more scalable way of looking at these analyses. And so, for example, we know that there are taxa that just have wacky 16S sequences and will never be picked up with our typical 16S primers. And so their efficiency is essentially zero. So we can have true zero efficiency taxa, but again, estimating these efficiencies is not, I think, a scalable approach here. Question from Curtis. Thanks so much for calling in, Curtis. Can I clarify the difference between concluding these measures never work versus these measures sometimes don't work? This is important in order to avoid sounding like the sky is falling, since there are plenty of good microbial community studies out there that do have sufficiently accurate relative abundance or diversity results based on internal controls follow-up validation. But of course, there are also plenty of bad ones too, and it's important in these tricky settings where cautions like these are important. Yeah, so I would definitely acknowledge that there's a lot of open work here and a lot of open questions. So the result from the Brooks and co-authors data, as well as the Costeyer and co-authors data that we looked at in the paper, is relatively clean compared to what we see in sort of wild microbial communities. We're doing an experiment at the moment investigating the effect of, for example, host DNA on these efficiencies. That's going to be an important factor. So I guess all I'm trying to say here is that I'm taking a lot of care in interpreting relative abundance data. I think that's not care that I often see being taken in the literature. And so maybe rather than saying, I guess I don't want to be up here and saying that the sky is falling, relative abundance data is garbage. We can't learn anything from quantitative microbiome data, as much as I would love everyone to think about the potential for differential efficiency of observing taxa to propagate through your analyses, and how robust an analysis and a conclusion is to such a data generating model. That's a terrific question. I don't want to sound like this is, none of this data is worth anything, but I'm happy to follow up on that, and thanks for the question. Yeah, and it sounds like Curtis is on board with the, remember that relative abundances are often pretty wonky for technical reasons, is a good, maybe more optimistic paradigm. But I do hope that work that we're doing is moving us in the direction of true calibration to some true protocol. So watch this space. Another question, more robust methods to replace estimation methods. Which methods should we incline ourselves to apart from validating qPCR plus 16S? I would say that is work in progress. We have not, there's some, I have some preliminary evidence that I haven't spoken about today, that maybe centered log ratio based analyses are more robust to these sorts of biases than looking at relative abundance data. As someone who's spent a lot of time developing diversity estimators and also relative abundance models, nothing pains me to say that more than that. But I would say, yeah, watch this space. This is sort of ongoing development, and we want to make sure, sort of going back to Curtis's point, that we want to make sure that what we're recommending in terms of tools as a result are validated in a lot of different systems, because we don't want to be only focusing on human gut and leaving out different types of analyses that would be more applicable in maybe vaginal microbiome or oral microbiome contexts. So yeah, ongoing work, and watch this space. I'm sorry I don't have anything more concrete for you today.
Okay, great. So I think that's it.